Hello to all of you. We are going to discuss chapter number 10 now. So, chapter number 10 is basically about mutations and cancer. We have discussed a lot of things before already because it's a course on cancer, but I'm going to go over some of the important things. So, you guys have already understood what cancer disease is and how mutations are responsible for causing cancer, and you know what mutations are. They are sudden change in DNA, and it's a change in the nitrogenous bases. And we have already discussed about the proto-oncogenes and the tumor suppressor genes in our previous chapters. So basically, cancer is a disease of unregulated division. So there are two very important genes called BRCA1 and BRCA2, and they're located on chromosome number 17 and 13. And it has been found that these genes, they are basically mutated in the breast cancer. So basically, this is chromosome number 17. It has a gene BRCA1, and chromosome number 13. It has a gene BRCA2. So these genes are mutated in basically breast and the ovarian cancer. So people who have mutations in these genes, they have a higher risk of uh, getting the developing the cancer. So this bar graph shows you that uh, this is the incidence rate of the breast cancer, and this is the incidence rate of the ovarian cancer in the who have BRCA1 and BRCA2 mutations. I have also provided you with a lot of videos for this chapter. Please watch them. They will help you understanding more about this topic. So basically, how do mutations arise? There are two kinds of mutations. They can be inherited mutations or they can be acquired mutations. Inherited mutations are those mutations which a person gets from the family, which means there are some mutations which are there in the genes. So cancer is basically a genetic disorder. It's basically an interplay of genes as well as some of the environmental factor, but genes play a very big role in cancer. So, so the acquired genes are those genes which a person acquires during the lifetime. So that can be because of environmental things. If you're eating something wrong, or if you are exposed to an environment where there is a lot of radiation. So these are all, or your eating habits are not correct. You are drinking alcohol. You are getting exposed to a lot of smoke. So these all reasons are responsible for the acquired mutations, the mutations which a person acquires during the lifetime. So in the breast cancer, around 90 to 95% are the acquired mutations that are responsible for cancer, and there are about 2 to 4.5% of the inherited mutations in BRCA1 and BRCA2, and these are 3 to 5% inherited mutations other than BRCA1 and BRCA2. And this is the incidence rate in all the other cancers. 90 to 95% is because of the acquired mutations, and 5 to 10% is responsible for the 5 to 10% are the inherited mutations which are responsible for cancer. So how do mutations arise? So mutation is a sudden change in DNA. So how do they arise? They can arise because of the environmental factors like smoking, sunburn, etc. Or they can also be arising because of the mistakes which are happening during DNA replication. So DNA replication is a process, as you know, we have already talked about that the two strands are separate, and each strand acts as a template strand, and on the template strand a daughter strand is made. So when the daughter strand is being made, at that time there is a possibility that if suppose a wrong nucleotide um, is incorporated, that can result in the mutations. So this is all talking about the inherited mutations, the mutations which we acquire from the families which are there in our genes. And this is talking about all the environmental things like UV radiation, X-rays, pollution, smoking, alcohol, charred or the like the barbecue meat and all that, that can also cause cancer. Viruses, cellular processes, some of the cellular reactions, they also are responsible for mutations. So, mutant is something, a chemical or a physical agent that can damage DNA by changing the nucleotide sequence. And carcinogen is something like cancer. So, most carcinogens are the mutagens. So, basically, how do mutations arise and how they affect? So, you can see this is the BRCA1 gene. So this is a normal BRCA1 gene. So this is this is the DNA, you can see, and this DNA codes for the mRNA, and then the mRNA codes for the amino acids, and these amino acids they code for a protein. So this is a normal BRCA1 gene. So you can see this is the DNA sequence, the mRNA sequence, and this is the sequence of the amino acids, and this is a normal BRCA1 gene. So suppose there is so in this basically one of the mutation which can take place in BRCA1 gene is the insertion frame shift. So there is the insertion of the nucleotide with a nitrogenous base thiamine. So there is an insertion of this here, and as a result of that, there is an insertion of an A adenine in the mRNA. So you can see here, this is a normal mRNA. It is AUG, CCG, then UCG. So because of the incorporation of the T here, there is the incorporation of A here. And after this, the whole sequence of the whole order of the amino acid after this changes. Okay. So you can see the order of amino acids is methionine, proline, serine, aspartic acid, then the glycine. Now after this methionine, proline, after this everything is changed. Okay, and the protein which is formed is different from the normal proteins, and this is the non-functional BRCA1 gene. So this is the impact of a mutation. So now we're going to talk about the mutations. How do the mutations basically in the oncogenes as well as how do the mutations basically impact the cell division, and how do they cause the cancer? So basically this is our cell cycle. This is the phases through which our cell passes during the life of the cell. So this is all is what is interphase. Okay, so this is G1, S and G2. So this all phase is the interphase. Followed by interphase is the mitotic phase, in which the cell divides. This we talked about in the last lecture. So at every phase, when each phase is changing, going from G1 to S, from S to G2, and then entering into the mitotic phase. At every phase, there are some proteins which actually take care if everything is going correct in the cell cycle. Like for example, when the cells are passing from G1 to S phase, at this point, when the cells are going to S phase, the DNA has to be duplicated. So when the cells are transitioning from G1 to S phase, there are some tumor suppressor proteins here, which are going to check if there are some proteins and they're tumor suppressor proteins. They're going to check if there is any damage to the DNA. If at this point the DNA is damaged, then because in the S phase the DNA has to be duplicated. So in the S phase the DNA has to be duplicated, and if there is a damage in the DNA, if the DNA is damaged, which means that that DNA should not be duplicated. So what's the function of tumor suppressor protein which is present here? So the function of this protein is to check if there is any damage to the DNA. If there is a damage to the DNA detected, then the cell will be given time to to make the repair. And if the repair is beyond the level that the cell can make that repair, that cell has to then die, and that is called apoptosis. So if the cell can correct the damage or repair the damage, then it is fine. But if the damage cannot be repaired, then that cell has to die by the process of apoptosis. So this is the way this is the way how cell cycle is regulated. So there are at every phase transition these are called checkpoints. So when the cell is going from G1 to S, there is a checkpoint. When the cell is going from S to G2, there is a checkpoint. So at every checkpoint there are some proteins which check if everything is going fine in the cell. Only then the cell should proceed to the next level. So at G1 to S checkpoint, it is going to be checked if there is any damage to the DNA. When the cell is going from S to G2, it is going to be checked if the DNA is duplicated. When the cell is going from G1, G2 to mitotic phase, I mean in the mitotic, sorry, when the cell is actually in the mitotic phase, it is going to be checked in the anaphase. There is a separation of the sister chromates. It's going to be checked if the chromosomes they are aligned properly, if the chromosomes are aligned properly for the chromatid separation. So at every step the cell cycle is very well regulated. But what happens if the proteins which are present at every checkpoint, if those proteins get mutated? If those proteins they get mutated, then they become dysfunctional. So if a tumor suppressor protein becomes taking care of the cell, if there is any damage to the DNA, if that protein gets mutated, then it will not be able to perform the function. So as a result of that, what will happen will be, if there is any damage to the DNA, the cell will keep on multiplying. So which means that it will lead to the growth of abnormal cells, and that is what is the disease called cancer, when there is an abnormal growth of the cells. Okay, so this is what I just said. So this I am not going to talk much, because we have already talked about what are proto-oncogenes and how do they get converted into oncogenes. So proto-oncogenes are those genes which are present in our cells. So proto-oncogenes are those genes which are present in our cells, and they are the one which are basically involved in the normal division of the cells. So mutations takes place in these proto-oncogenes, and these proto-oncogenes get converted into oncogenes, and as a result of that there is a abnormal cell division. And we have another set of genes which are called tumor suppressor genes, and these are the guardians of our genome, and they take care if everything is going well with the cell cycle. So these tumor suppressor genes, they also get mutated, and when these get mutated, that can also lead to the abnormal all various times. I'm going to spend much time on this. So this is all we have discussed. So cancer can be basically when the cancer is so benign tumor, basically it can be a non-cancerous tumor. Usually when the cancer is confined to the place of its origin, at that point it cannot it can be removed and the cancer doesn't spread out. In that situation we call the cancer to be benign. When the cancer is confined to one organ, the cancer cells can be removed from that organ, and the cancer does not spread out. So in that situation it is called benign tumor. But when the cancer spreads out and goes to distant organs, then we call the tumor is malignant. And metastasis is the process by which there is a spread of the cancer cells. How cancer can be treated? There are various ways of treatment of cancer. Chemotherapy, so which basically use some chemicals. Radiation therapy, using radiation. Targeted therapy is that therapy which basically targets the cancerous cells, which targets the specific cancer cells. Therapy is making use of the basically it's a very current area, immunotherapy. It's basically using your own immune system to attack the cancer cells. How we can reduce the risk of cancer? There are various ways by which we can reduce the risk of cancer. First of all, Don't smoke or chew tobacco because you are exposing your cells to a lot of chemicals, and these chemicals can go and damage your DNA. Eat healthy and maintain a healthful weight. Get vaccinated. Yes, it's very important because there are some cancers, and the cause of those cancers are viruses. Avoid harmful environments like where there is a lot of radiation and all that. Don't be there. Screen regularly for cancer. So there are various tests which are done. There's a pap smears done, mammograms done. They're all screening tests, so that should be done. And get tested for the hereditary cancers. So basically, um, there are some companies who can go and check your DNA, and they can check if you are susceptible to cancer, and basically they go and look for some mutations in our DNA. So this is the way we can reduce the risk of cancer. So that's all about this chapter. Any question, let me know, and please go over all the material which I posted on D12. I posted various videos. Please watch them and take in-class quiz. And if you have any question, please